Tonight's lecture is over Chapter 6, Metabolic Pathways and Cellular Respiration. We sometimes use the word respiration to mean breathing, while respiration on the organismal level should not be confused with cellular respiration. The two processes are closely related. Cellular respiration requires a cell to exchange two gases with its surroundings. A cell takes in oxygen in the form of the gas O2. It gets rid of waste in the form of the gas carbon dioxide, or CO2. Breathing results in the exchange of these same gases between your blood and the outside air. Oxygen present in the air you inhale diffuses across the lining of your lungs into your bloodstream, and the CO2 in your bloodstream diffuses into your lungs and exits when you exhale. Internal combustion engines, like the ones found in cars, use oxygen via the air intakes to break down gasoline. A cell also requires O2 to break down its fuel. Cellular respiration, which is a living version of internal combustion, is the main way that chemical energy is harvested from food and converted to ATP energy. Cellular respiration is an aerobic process, which is just another way of saying that it requires oxygen. Putting all this together, we can now define cellular respiration as the aerobic harvesting of chemical energy from organic fuel molecules. There are three metabolic pathways involved in cellular respiration, and they are glycolysis, the citric acid cycle, and the electron transport chain. As I said, cellular respiration is an example of a metabolic pathway. That means it is not a single chemical reaction, but a series of reactions. A specific enzyme catalyzes each reaction in a metabolic pathway. More than 24 reactions are involved in cellular respiration. We can group them into the three main metabolic stages, as I've already mentioned: glycolysis, the citric acid cycle, and the electron transport chain. Moving on, humans and other animals depend on plants to convert the energy of sunlight to the chemical energy of sugars and other organic molecules we consume as food. This is accomplished via photosynthesis. Photosynthesis uses light energy from the sun to power a chemical process that builds organic molecules. But we depend on plants for more than our food. You're probably wearing clothing made of another product of photosynthesis, cotton. Most of our homes are framed with lumber, which is wood produced by photosynthetic trees. Plants and other autotrophs are organisms that make all their own organic matter, including carbohydrates, lipids, proteins, and nucleic acids, from nutrients that are entirely inorganic, such as carbon dioxide from the air and water and minerals from the soil. In contrast, humans and other animals are heterotrophs, organisms that cannot make organic molecules from inorganic ones. Therefore, we must eat organic material to get our nutrients. Most ecosystems depend entirely on photosynthesis for food. For this reason, biologists refer to plants and other autotrophs as producers. Heterotrophs, in contrast, are consumers because they obtain their food by eating plants or by eating animals that have eaten plants. We animals and other heterotrophs depend on autotrophs for organic fuel and for the raw organic materials we need to build our cells and tissues. So in these next two slides, we're going to contrast the cells of plants, or producers, autotrophs, with that of animals, heterotrophs, or consumers. So in this slide, we see a plant cell. Notice the cell wall, notice the mitochondria, and notice the chloroplast. So the chloroplast is going to be the site of photosynthesis, and the mitochondria is going to be the site of cellular respiration. And here we see the animal cell, and what the animal cell lacks is the cell wall and the chloroplast. However, the animal cell does have mitochondria that produces energy via the cellular respiration process. Moving on, the chemical ingredients for photosynthesis are carbon dioxide, a gas that passes from the air into a plant via tiny pores, and water, which is absorbed from the soil by the plant's roots. Chloroplasts in the cells of the leaves use light energy to rearrange the atoms of these ingredients to produce sugars, most importantly glucose, and other organic molecules. A byproduct of photosynthesis is oxygen gas. Both animals and plants use organic products of photosynthesis as sources of energy. A chemical process called cellular respiration harvests the energy that is stored in sugars and other organic molecules. Cellular respiration uses oxygen to help convert energy extracted from organic fuel to another source of chemical energy called ATP. Cells expend ATP for almost all of their work. In both plants and animals, the production of ATP during cellular respiration occurs mainly in the organelles called mitochondria. It should be noted that the waste products of cellular respiration are carbon dioxide and water, the very same ingredients used for photosynthesis. Plants store chemical energy via photosynthesis and then harvest this energy via cellular respiration. However, plants usually make more organic molecules than they need for fuel. This photosynthetic surplus provides the organic material for the plant to grow. It is also the source of food for humans and other consumers. It should also be noted that plants perform both photosynthesis to produce fuel molecules and cellular respiration to burn them, while animals perform only cellular respiration. Analyze nearly any food chain, and you can trace the energy and raw materials for growth back to solar-powered photosynthesis. So getting back to cellular respiration, a common fuel molecule for cellular respiration is glucose, which is a 6-carbon sugar with the formula C6H12O6. Here is the overall equation for what happens during cellular respiration. We start with the glucose molecule and 6 oxygen. The series of arrows indicates that cellular respiration consists of many chemical steps, not just a single chemical reaction. Remember, the main function of cellular respiration is to generate ATP for cellular work. In fact, the process can generate up to 38 ATP molecules for each glucose molecule that is consumed. And we see after the series of arrows, or the series of chemical reactions, we end with 6 carbon dioxide, 6 waters, and then ATP, or energy. Notice that cellular respiration also transfers hydrogen atoms from glucose to oxygen, forming water. That hydrogen transfer turns out to be key as to why oxygen is so vital in the harvest of energy during cellular respiration. Oxygen is what we call the final electron acceptor. And that brings us to the role of oxygen in cellular respiration. So in tracking the transfer of hydrogen from sugar to oxygen, we are also following the transfer of electrons. The atoms of sugar and other molecules are bonded together by shared electrons. During cellular respiration, hydrogen and its bonding electrons change partners from sugar to oxygen, forming water as a product. So chemical reactions that transfer electrons from one substance to another substance are called oxidation-reduction reactions, or redox reactions for short. The loss of electrons during a redox reaction is called oxidation. Glucose is oxidized during cellular respiration, losing electrons to oxygen. The acceptance of electrons during a redox reaction is called reduction. Adding electrons is called reduction. Negatively charged electrons added to an atom reduce the amount of positive charge of that atom. Oxygen is reduced during cellular respiration by accepting electrons from glucose. An easy way to remember this is by the acronym OIL-RIG. Oil rig stands for oxidation is a loss and reduction is a gain. But water isn't the only thing produced when hydrogen and its bonding electrons change patterns from glucose to oxygen. Energy is also released. Why does electron transfer of oxygen release energy? In redox reactions, oxygen is a quote-unquote electron grabber. An oxygen atom attracts electrons more strongly than almost any other type of atom. When electrons move along with hydrogen from glucose to oxygen, it is as though they are falling. They are not really falling in the sense of an apple dropping from a tree. However, in both cases, potential energy is unlocked. Instead of gravity, it is the attraction of electrons to oxygen that causes the quote-unquote fall and energy release during cellular respiration. The reaction between hydrogen gas and oxygen gas producing water releases a large amount of energy as the electrons of hydrogen fall into their new bonds with oxygen. This reaction is actually quite explosive, and it would be difficult for a cell to capture such a burst of energy and put it to useful work. As you'll see shortly, cellular respiration is a more controlled fall of electrons, more like a stepwise cascade of electrons down an energy staircase. Instead of liberating food energy in a burst, cellular respiration unlocks chemical energy in smaller amounts that cells can put into productive use. But let's take a closer look at the path that electrons take on their way from glucose to oxygen. The first stop is a positively charged electron acceptor called NAD+, which stands for nicotinamide adenine dinucleotide. The transfer of electrons from organic fuel, food in our case, to NAD+, reduces the NAD+ to NADH. The H represents the transfer of hydrogen along with the electrons. In our staircase analogy, the electrons have now taken one baby step down in their trip from glucose to oxygen. The rest of the staircase consists of an electron transport chain. Each link in an electron transport chain is actually a molecule, usually a protein, in a series of redox reactions. Each member of the chain first accepts and then donates electrons. With each transfer, the electrons give up a small amount of energy that can be used indirectly to generate ATP. The first molecule of the chain accepts electrons from NADH, thus the NADH carries the electrons from glucose and other fuel molecules and deposits them at the top of the electron transport chain. The electrons cascade down the chain from molecule to molecule like an electron bucket brigade. The molecule at the bottom of the chain finally drops the electrons to oxygen. The oxygen also picks up hydrogen, forming water. The overall effect of this transfer of electrons during cellular respiration is a downward trip for electrons from glucose to NADH to an electron transport chain to oxygen. During the stepwise release of chemical energy during the electron transport chain, our cells make most of their ATP. It is actually oxygen, the quote-unquote electron grabber, that makes it all possible by pulling electrons down the transport chain from fuel molecules. Oxygen functions somewhat like a gravity well, pulling objects downhill. This is how the oxygen we breathe functions in our cells and why we cannot survive more than a few minutes without it. Viewed this way, drowning is deadly because it deprives cells of the final electron acceptor, oxygen, needed to drive cellular respiration. So this slide gives us an overview of cellular respiration. Cellular respiration is an example of a metabolic pathway. That means that it is not a single chemical reaction, but a series of reactions. A specific enzyme catalyzes each reaction in the metabolic pathway. More than 24 reactions are involved in cellular respiration. We can group them into three main metabolic stages: glycolysis, the citric acid cycle, and the electron transport chain. During glycolysis, a molecule of glucose is split into two molecules of a compound called pyruvic acid. The enzymes for glycolysis are located in the cytoplasm. The citric acid cycle, which can also be called the Krebs cycle, completes the breakdown of glucose all the way to carbon dioxide, one of the waste products of cellular respiration. The enzymes for the citric acid cycle are dissolved in the fluid within the mitochondria. Glycolysis and the citric acid cycle generate a small amount of ATP directly. They generate much more ATP indirectly via redox reactions that transfer electrons from fuel molecules to NAD+, forming NADH. The third stage of cellular respiration is the electron transport chain. Electrons are captured from food by the NADH formed in the first two stages of the quote-unquote fall down the electron transport chains to oxygen. The proteins and other molecules that make up the electron transport chains are embedded within the inner membrane of the mitochondria. Electron transport from NADH to oxygen releases the energy your cells use to make most of their ATP. So in this slide, we'll discuss glycolysis in a little more detail. The word glycolysis means the splitting of sugar. That is exactly what happens. During glycolysis, a 6-carbon glucose molecule is broken in half, forming two 3-carbon molecules. Notice that the initial split requires an energy investment of 2 ATP molecules per glucose. The 3-carbon molecules then donate high-energy electrons to NAD+, the electron carrier forming NADH. In addition to NADH, glycolysis also makes 4 ATP molecules directly when enzymes transfer phosphate groups from fuel molecules to ADP. Glycolysis thus produces a net of 2 molecules of ATP per molecule of glucose. This fact will become important during our discussion of fermentation. What remains of the fractured glucose at the end of glycolysis are 2 molecules of pyruvic acid. The pyruvic acid still holds most of the energy of glucose, and that energy is harvested in the second stage of cellular respiration, the citric acid cycle. Moving on, pyruvic acid, which is the fuel that remains after glycolysis, is not quite ready for the citric acid cycle. Remember that each molecule of glucose produces 2 molecules of pyruvic acid. The pyruvic acid must be quote-unquote prepped or converted to a form the citric acid cycle can use. First, pyruvic acid loses a carbon as CO2. This is the first of the waste products that we've seen so far in the breakdown of glucose. The remaining molecules, each with only 2 carbons left, are called acetic acid, the acid that's in vinegar. Oxidation of the fuel generates NADH. Finally, the acetic acid is attached to a molecule called coenzyme A to form acetyl-CoA. The CoA escorts the acetic acid into the first reaction of the citric acid cycle. The CoA is then stripped and recycled. And that brings us to the citric acid cycle. The citric acid cycle finishes extracting the energy of sugar by breaking down the acetic acid molecules all the way down to carbon dioxide. Acetic acid joins a 4-carbon acceptor molecule to form a 6-carbon product called citric acid, for which the cycle is named. For every acetic acid molecule that enters the cycle as fuel, 2 carbon dioxide molecules eventually exit as a waste product. Along the way, the citric acid cycle harvests energy from the fuel. Some of the energy is used to produce ATP directly. However, the cycle captures much more energy in the form of NADH and a second closely related electron carrier called FADH2. All the carbon atoms that entered the cycle as fuel are accounted for as carbon dioxide exhaust. and the 4-carbon acceptor molecule is recycled. We have tracked only one acetic acid molecule through the citric acid cycle here, but since glycolysis splits glucose in two, the citric acid cycle actually turns twice for each glucose molecule that fuels a cell. And that brings us to the third stage of cellular respiration, which is the electron transport chain. The molecules of the electron transport chain are built into the inner membranes of the mitochondria. An electron transport chain functions as a chemical machine that uses the energy released by the quote-unquote fall of electrons to pump hydrogen ions across the inner mitochondrial membrane. This pumping causes ions to become more concentrated on one side of the membrane than the other. Such a concentration gradient stores potential energy. The energy stored by the electron transport behaves something like the elevated reservoir of water behind a dam. There is a tendency for hydrogen ions to gush back to where they are less concentrated, just as there is a tendency for water to flow downhill. The inner membrane is analogous to the dam and temporarily restrains hydrogen ions. The energy of dammed water can be harnessed to perform work. Gates in the dam allow the water to rush downhill, turning giant turbines as it goes. The spinning turbines perform work that can be used to generate electricity. Your mitochondria have structures that act like turbines. Each of these miniature machines, called an ATP synthase, is constructed from a protein built into the inner mitochondrial membrane adjacent to the proteins of the electron transport chain. Moving on, this figure on this slide shows a simplified view of how energy previously stored in NADH and FADH2 can now be used to generate ATP. NADH and FADH2 transfer electrons to an electron transport chain. The electron transport chain uses this energy supply to pump hydrogen ions across the inner mitochondrial membrane. Oxygen then pulls electrons down the transport chain. The hydrogen ions concentrated on one side of the membrane rushes back downhill through the ATP synthase. This action spins a component of the ATP synthase, just as water turns the turbines in a dam. The rotation activates parts of the ATP synthase molecule to attach phosphate groups to ADP molecules to generate ATP. And moving on to the next slide, we see the ADP being converted into ATP in this cartoon. The poison cyanide produces its deadly effect by binding to one of the protein complexes in the electron transport chain. When bound there, cyanide blocks the passage of electrons to oxygen. This blockage is like turning off a faucet. The electrons cease to flow through the pipe. As a result, no hydrogen ion gradient is generated and no ATP is made. Thus, cells stop working and the organism dies. Cyanide was the lethal agent in an infamous case of product tampering, the Tylenol murders of 1982. 7 people in the Chicago area died after ingesting Tylenol capsules that had been laced with cyanide. And this next slide shows us an overview of the electron transport chain. And this slide shows us an overview of cellular respiration. Moving on, we have seen that glucose can provide the energy to make the ATP our cells use for all of their work. The importance of glucose is underscored by the severity of diseases in which glucose balance is disturbed. Diabetes, which affects over 20 million Americans, is caused by an inability to properly regulate glucose levels in the blood due to problems with the hormone insulin. If left untreated, a glucose imbalance can lead to cardiovascular disease, coma, and even death. But even though we have concentrated on glucose as the fuel that is broken down during cellular respiration, respiration is a versatile metabolic furnace that can burn many other kinds of food molecules. The diagram on this slide shows the metabolic route for the use of carbohydrates, fats, and proteins as fuel for cellular respiration. Taking cellular respiration apart to see how all the molecular nuts and bolts of its metabolic machinery work, it's easy to lose sight of its overall function, that is, generating up to 38 molecules of ATP per molecule of glucose, and the actual number can vary by a few. Glycolysis and the citric acid cycle contribute to ATP by direct synthesis. All of the rest of the ATP molecules are produced by the ATP synthase machines that power the fall of electrons from food to oxygen. The electrons are carried from the organic fuel to the electron transport chains by NADH and FADH2. Each electron pair dropped down the transport chain from NADH can power the synthesis of up to 3 ATP. Each electron pair transferred to an electron transport chain from FADH2 is worth up to 2 ATP. Now we'll see what happens when cells harvest food energy without the help of oxygen. Although you must breathe to stay alive, some of your cells can work for short periods without oxygen. This anaerobic harvest of food energy is called fermentation. When you walk between classes, your leg muscles require a constant supply of ATP, which is generated by cellular respiration. To keep this process going, blood provides your muscle cells with enough oxygen to keep electrons falling down the transport chains in your mitochondria. But if you start to run because you're late for class, your muscles are forced to work under anaerobic conditions. That's because they are spending ATP at a rate that outpaces your bloodstream's delivery of oxygen from your lungs to your muscles. After functioning anaerobically for about 15 seconds, muscle cells will begin to generate ATP by the process of fermentation. Fermentation relies on glycolysis, the same metabolic pathway that functions as the first stage of cellular respiration. Glycolysis does not require oxygen, but does produce 2 ATP molecules for each glucose molecule broken down to pyruvic acid. That isn't very efficient compared to the 38 or so ATP molecules each glucose generates during the cellular respiration, but it can energize your leg muscles long enough for you to make it to class. However, your cells will have to consume more glucose fuel per second since so much less ATP per glucose molecule is generated under anaerobic conditions. To harvest food energy during glycolysis, NAD+ must be present as an electron acceptor. This is no problem under aerobic conditions because the cell regenerates NAD+ when NADH drops its electron cargo down the electron transport chains to oxygen. However, this recycling of NAD+ cannot occur under anaerobic conditions because there is no oxygen to accept the electrons. Instead, NADH disposes of electrons by adding them to the pyruvic acid produced by glycolysis. This restores NAD+ and keeps glycolysis working. The addition of electrons to pyruvic acid produces a waste product called lactic acid. The lactic acid byproduct is eventually transported to the liver, where liver cells convert it back to pyruvic acid. Exercise physiologists, those are biologists who study the effects of exercise on the body, have long speculated about the role that lactic acid plays in muscle fatigue. Moving on, our muscles cannot function by lactic acid fermentation for very long. However, the 2 ATP molecules produced per glucose molecule during fermentation is enough to sustain many microorganisms. We have domesticated such microbes to transform milk into cheese, sour cream, and yogurt. These foods owe their sharp or sour flavor mainly to lactic acid. The food industry also uses fermentation to produce soy sauce from soybeans, to pickle cucumbers, olives, and cabbage, and to produce meat products like sausage, pepperoni, and salami. Yeast, a microscopic fungus, is capable of both cellular respiration and fermentation. If you keep yeast cells in an anaerobic environment, they are forced to ferment sugars and other foods to stay alive. When yeast ferment, they produce ethyl alcohol, a waste product, instead of lactic acid. This alcoholic fermentation also releases carbon dioxide. For thousands of years, humans have put yeast to work producing alcoholic beverages such as beer and wine. And as every baker knows, the CO2 bubbles from fermenting yeast also cause bread dough to rise. The alcohol produced in fermenting bread is released during baking. So let's summarize tonight's lecture. After tonight's lecture, you should know the 3 major pathways of cellular respiration. You should know what the major products are of each. You should know what organelle is needed for aerobic cellular respiration. You should know how much ATP is made during each pathway and in total for aerobic cellular respiration. You should know what other molecules we can use to make ATP. You should know what the 2 types of fermentation are. You should know what the products of the different types of fermentation are. You should also know where we see fermentation in our everyday lives. And you should know why it is so important that our cells have mitochondria and that we get the cells enough oxygen.